The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 45 of the eLife podcast, which is produced by The Naked Scientists. I'm Chris Smith. This month, how insects turn leaves into amplifiers so they can make even more noise, new insights into the brain basis of post-traumatic stress disorder, and the initiative to map every cell type in the human body. But first, this winter we've seen very high levels of respiratory infection, which have caused severe outbreaks in hospitals and care home settings. Transmission tends to occur in these environments because individuals are cared for at high density. And the way institutions control infections under these circumstances is by strict policies that isolate and even exclude cases. That's us humans, but there are other classes of animals that also live in high density, and they've developed ingenious infection control strategies of their own. Chris Pull has been looking at what ants do.
We know that social insects, so that's ants, bees, wasps and termites, have evolved collective disease defences to try and control epidemics in their colonies. But a lot of the work so far has looked at how they prevent infections. So, for example, they groom one another and they use antimicrobial disinfectants to prevent individuals which come into contact with pathogens from actually contracting an infection. But what we wanted to know is how they actually prevent successful infections from spreading. So in cases where these sort of first line defenses fail to prevent disease, what can a colony do to prevent the infection spreading to others?
How do they know that they have an outbreak situation in the first place?
So what we've been able to show through our research using chemical analyses is that they can actually smell when another individual is sick. So we've shown that sick individuals, when they have an infection and when you also inject them with an immunoliciter, increase cuticular hydrocarbons, and this attracts the attention of ants in the colony and triggers a response.
So this is like ant BO, isn't it? These cuticular hydrocarbons that they can sniff on each other.
Yes, exactly. So they use them typically to tell if you're a member of the colony or not. But we've been able to show now that they actually also change them in response to infection, and that can actually tell others who's sick.
So what is the situation when they pick up that this chemical trace or chemical signature of disease is there? How do they respond?
It's quite interesting. So they sort of have this multi-component behavior. So we were looking at infections in pupa, and the pupae are the sort of developmental stage in between a larvae and an adult ant. They're going through metamorphosis, and they're encased in these silk cocoons. And what we found is that upon detecting an infection, the ants will actually break open this silk cocoon, and then they start biting the infected pupa, and then they spray poison, which is made up of formic acid and acetic acid, from a gland at the end of their abdomen. And this ensures that the fungus or the pathogen growing inside the infected brood can't grow anymore. So the acids actually seem to kill this fungus, which is inside the body of the pupa. And it seems like they do all this because the poison itself can't actually penetrate into the body of the pupa unless the cocoon is removed and unless they make these holes in the body of the pupa itself.
And can you demonstrate that this really does mitigate or curtail the spread? So in other words, if you abolish this behaviour, it would be curtains for the colony.
Yeah, so we've actually been able to show by mimicking a situation where they failed to detect and destroy these infections. So we simply kept ants with an infectious pupa. 40% of these small groups of ants actually contracted the infection and became infectious themselves. And you can imagine that in a full colony setup, that can very quickly lead to a sort of huge mass breakout of this disease. But by performing these behaviors, we actually saw that there was zero disease transmission.
Do other social insects that have similar problems deal with it the same way, or do they have a different strategy?
So we do see different strategies. So in honeybees, because they live in these hives and they forage on the wing, what they can do is to simply take diseased brood out of the nest, fly away a few hundred meters and just drop it somewhere in the vegetation. And because they forage on plants and they forage for wide distances around the colony, the chances that they re-encounter that infectious corpse are really low. The termites, on the other hand, what they do is actually eat their dead. So they live encased in these sort of pieces of wood which are rotting away. And for them, it's hard to remove things from the wood because they really live inside it. So what they tend to do is to actually eat their diseased individuals. But at the end of the day, they all use more or less a similar strategy. So they're all trying to detect very early these infections and to either remove or to destroy or to eat them before they have the chance to become infectious.
That's quite an undertaking strategy, isn't it? Actually
Yeah.
eating your dead. But how do you think this evolved in the first place? Because it's quite a complicated behaviour, isn't it? It involves the ability to do chemical detection and recognition, and then to have evolved a strategy that is itself successful in mitigating the threat.
Yeah, so we think that these behaviors have evolved because social insect colonies are like a superorganism. So they behave and they reproduce like a single organism in itself. And in a way, they're very similar then to a multicellular body, like a frog or a human being. And in the same way, when a human has an infected cell in its body, you have this immune reaction to remove that infected cell. And we see then common processes in sort of multicellular organisms and these sort of superorganismal insect societies. And we think that common evolutionary processes were at play during the evolution of both multicellular organisms and superorganisms, and being able to basically detect and remove elements which might harm the entire organism in itself were necessary sort of prerequisites, or at least were necessary to evolve in order to ensure that like you have the survival of the whole over its parts.
Chris Paul, he's at Royal Holloway. Have a listen to this. And now, keep the volume of what you've just heard in mind and compare it with this. Hopefully you'll agree that the second set of sounds were a lot louder. But we've not manipulated the recording at all. Instead, the insect that made that sound did it itself by building its own amplifier from a leaf. And Natasha Mahatre has discovered how.
So these creatures are called tree crickets. They're about 1 centimetre in length. They're really beautiful. I mean, most of us see crickets from the pet store, which look kind of brown and a bit cockroach-like. Tree crickets are really pretty. They have translucent wings. They have long antennae, they're small green things. And there's tree cricket species more or less all over the world. The ones that we're working on come from India. All tree crickets do this really amazing thing, which is that they sing to attract mates. They put up their wings, which are resonant, rub them together, set them into vibration, and produce really beautiful tonal sounds. That's what you can hear most summer nights. These sounds are heard by the females, who can use them to identify males of their own species from other males and find the males. These tree crickets also do this amazing other thing, which is that they make an aid that helps them be louder than they would be on their own.
Really? What do they do?
They make a thing called a baffle. A baffle is, for a tree cricket, very simply, a hole cut in a leaf that they sing from. So they place their wings directly against this hole and sing from inside it, and this makes them louder.
Is that a bit like you see people walking around in the old days on Hollywood sets with a megaphone, which was basically a cone with the end chopped off, and they shout into it? Is it sort of doing something similar? They're creating an amplifier from a leaf to make themselves louder?
It's a bit similar, so I'll try and explain the physics as simply as I can. So if you can imagine the wings as a board that's vibrating back and forth, every time the board moves forward, it produces high pressure in front of it. And behind it, it produces low pressure. And this is essentially what sound is. It's changes in pressure. But what will happen is that the high pressure and the low pressure will meet at the edge of the board. And when they meet with each other, they'll cancel each other out. And this is something we call acoustic short circuiting. Now, the smaller the board is, the more of the sound that's produced is short-circuited. If you can somehow prevent these two, high pressure and the low pressure, from meeting each other, then you can prevent acoustic short-circuiting. And the more you can do this, the more loud the sound will be. And that's essentially what's happening when a cricket puts itself in the hole in a leaf. What happens is that the front face of the wing is effectively acoustically separated from the back face of the wing, and they end up being louder.
And how does the cricket find the leaf and the hole in the first place?
There's a big secret in that. So one of the things that we did was to see whether there are different baffle designs. So basically, what are the different sizes of leaves that are available to the cricket? What are the different sizes of holes they can make? Where do they put the hole? And it turned out, if you model this, you find that some baffles perform a lot better than others. And the baffle that performs the best is made with the largest leaf, with a hole that's exactly the size as the wings, and placed dead center in the leaf.
Right, so next question then. Given that that's the optimal solution, what proportion of the crickets actually opt for that? In other words, are they actively pursuing that solution, or do they arrive at that sometimes by chance?
Well, it depends on the situation, but when we did an experiment in which we gave 19 crickets the choice between a small leaf, which wouldn't make such a good baffle, and a large leaf, which would make a great baffle, 15 crickets made a baffle, and every single one of them made a baffle in the large leaf. They made a baffle in the large leaf with the optimally sized hole, and they got pretty close to the center. So all of the crickets seemed to know how to make an optimal baffle.
Now, given that crickets sing at night, they're therefore able to work out the size of a leaf, discriminate between leaves that are bigger and smaller, and then having worked out the size of the leaf and made a choice, they're then working out where the middle is. How on earth are they doing that?
They are doing that. We have no idea. There may be different ways in which they're doing this. One is to walk along the edge of the leaf. The other is to use these really beautiful long antennae that they have to touch the edges of the leaf to see if the edges are further away on one leaf than another one, and, sort of using them to center themselves in the middle of the leaf. But to be honest, the answer is we don't know how they're doing it yet.
And now that you've done these experiments and you can actually make the measurements, the sounds they're producing, how much louder is the baffled cricket compared with a non-baffled cricket? In other words, what sort of advantage, a sonic advantage, do they gain through doing this?
So between a cricket that's singing on its own and one in an optimal baffle, there's a 4 times change. So a cricket that's in an optimal baffle will be 4 times as loud as a cricket that's on its own.
This completely changes our view of insects, doesn't it? Because we mostly think of insects as sort of dumb automatons with a brain that's sort of hardwired to process inputs and generate outputs. But here you've got very simple organisms. They're making a sequence of decisions informed by their own measurements in order to achieve an outcome. And that's really quite striking.
Absolutely. It has been a long-held view that invertebrates are stereotyped, and vertebrates, mammals, birds, etc., are extremely clever. And we've been having to change this view. I mean, I think one of the first things that really jolted this was when they found that octopuses can make tools. They were making all sorts of tools. Now, if you think about it a little bit, an octopus is a mollusk. It's the same as a snail. So really, we can't hold on to this idea anymore that invertebrates are simpler animals. They're just different from us. And we're absolutely going to have to look very closely at how insects behave in order to be able to appreciate what's going on that's not immediately apparent on the surface.
And who would have thought that insects could be acoustics masterminds? That was Natasha Mhatre, and she's at the University of Toronto. You're listening to the eLife podcast with me, Chris Smith. Still to come, what's happening in the brain of a person with PTSD and the ambitious initiative to map every cell in the human body. But before that, every year, millions of women fall pregnant. It's a fact of life. But up to 10% of them will develop the condition preeclampsia, which is associated with dangerously high blood pressure that can even be life-threatening if it isn't managed promptly and appropriately. And although doctors are very good at doing that, we're still not entirely sure why the condition occurs at all, which makes predicting and preventing it much more difficult. Now, MIT scientist Katie Bezold-Lamb has found a way to produce a very similar manifestation in mice, which might give us new clues as to what's going on.
The placenta is a specialized organ that only occurs during pregnancy. It's created by the developing baby. It's the site of communication between mom and baby and the site of gas and nutrient exchange for the developing baby.
How does it actually form?
So the placenta is formed by invasive cells from the developing fetus that invade into the maternal uterus. These cells locate maternal spiral arteries and replace the underlying endothelium, widening these arteries to increase the amount of blood flow to the placenta, effectively increasing the amount of available nutrients and oxygen for the placenta to deliver to the baby. However, in a certain subset of pregnancies, this invasion of specialized cells called trophoblasts does not occur quite as effectively as it should. The spiral arteries aren't properly remodeled, so the amount of blood flow available is drastically decreased. And since these spiral arteries are thinner or the diameter is reduced, the mother is at risk for developing high blood pressure in pregnancy, which can lead to preeclampsia. Additionally, since there is a reduction in the amount of blood flow available, the baby is at risk for developing intrauterine growth restriction, where the baby is then small for its gestational age.
It's quite common, this, isn't it, preeclampsia? But do we actually know what causes it?
Absolutely. Preeclampsia is very common, but while it is generally understood that preeclampsia arises due to shallowly invading trophoblast cells, we don't actually know what regulates invasion of these cells and what goes wrong to cause preeclampsia. And so we sought to determine the mechanisms that regulate trophoblast invasion. And so we decided to study the inverted formin 2 protein, which is responsible for generating invasive structures in cells.
So what actually is this protein? Where does it come from and how does it act on a cell?
So inverted formin 2 is actually found in a wide variety of different cell types, but what its primary role is in the cell is to maintain the actin cytoskeleton. It has a unique ability to depolymerize actin filaments. This is important in invasion because cells need to be able to form actin-rich protrusions that allow cells to invade.
Right, so you've got this protein. It's made in these trophoblast cells that are doing the invading into the maternal arteries, and they're expressing heavily this protein, which can remodel the skeleton inside the cell, which will warp the cells, making them change shape and bend and twist and so on. So is your hypothesis then that this protein is in some way linked to the ability to invade, and if it wasn't being expressed correctly or in the right amounts, that might be why invasion fails or doesn't progress far enough down the artery, and that's what causes preeclampsia?
Yes, our hypothesis was that inverted formin 2 is necessary for trophoblast invasion, and that loss of inverted formin 2 would reduce spiral artery invasion and lead to preeclampsia. So to test this hypothesis, we used a culture method of extravillous trophoblasts in which we've knocked down inverted formin 2 and saw that these cells had a reduced capability of invasion.
What about in vivo, though? Because it's one thing to do this in a dish. It's a very different effect in vivo.
That's actually a really good question. So to understand the role of inverted formin 2 in vivo, we turned to a knockout mouse model. When we studied the pregnancies of these knockout mice, we found that the pregnant mice displayed new onset high blood pressure late in pregnancy, and that it wasn't until they delivered the pups and their placentas that blood pressure returned to normal. Additionally, we looked at the spiral artery remodeling in these INF2 knockout mice and found that there was a significant reduction in the number of spiral arteries in these knockout mice, suggesting that spiral artery remodeling in these mice is reduced. The pups in these INF2 knockout mice were significantly smaller than the wild-type counterparts.
So basically what you've managed to create is a very, very close mimic of what we see in human preeclampsia, because you've got animals that develop hypertension, they have intrauterine growth retardation, and all of the other negative consequences go away after the animals give birth, which is a very strong mimic for what we see in the human condition.
Yeah, it is a really interesting phenotype that does recapitulate several aspects of preeclampsia and intrauterine growth restriction. I do think we are a bit far from thinking about potentially treating preeclampsia, but this would provide an interesting method for screening people at increased risk for developing preeclampsia.
And it will be very interesting to see what that new model reveals about a very common human condition. Katie Beazold-Lamb there. People with post-traumatic stress disorder, or PTSD, experience harrowing flashbacks to frightening events and encounters from their past. Usually these are triggered by some sort of reminder of what happened to them originally, and these patients are also much more alert to potential threats than the average person. But what's changed in their brains to make them respond like this? Pearl Chu and Brooks Kincasus are at Virginia Tech, where they've been trying to find out.
Post-traumatic stress disorder, PTSD, is a debilitating psychiatric illness that often occurs after someone's experienced a dangerous or threatening event. And people with PTSD tend to overreact to unexpected reminders of the events and are often hypervigilant for danger. And so what we've done here is take new findings from computational neuroscience, from computer science, from statistics to try to fit models to understand what might be happening in PTSD.
So in essence, which bits of the brain are miswiring or misfiring in order to provoke the changes in behaviour you see in someone who's manifesting PTSD?
Yeah, that's exactly right.
And Brooke, how did you actually do that?
So we recruited 75 veterans who had served in Iraq and Afghanistan, and we brought them into the lab and placed them into a fMRI scanner. While we were monitoring the blood flow in their brain, we had them play a gambling task in which they were trying to evaluate and make decisions between two different options, one of which was better than the other.
So Pearl, why does a gambling task actually help to shed light in this instance?
Previous data suggest that PTSD may be a disorder of disrupted learning. So one of the symptoms of PTSD is hypervigilance. When you encounter something unexpected, people with PTSD tend to overreact. And a gambling task in the learning context lets us evaluate what parts of learning, what parts of the brain may be disrupted. The way we're able to quantify surprise in the context of a behavioral paradigm is people with and without PTSD are choosing between two stimuli or two ambiguous pictures and trying to learn which one is better, and then you see the outcome. And we can infer what your expectations were by how surprised you are by the outcome.
I get it. So essentially, you put them in the scanner, you give them a gambling task, and they learn, if I do this, I win, if I don't do that, I lose. Then you, flip the odds so that then they're losing when they don't expect to and they're surprised?
Yeah, well, every so often they'll get something that they're not expecting. And in those cases, we're studying how the brain is responding to those unexpected events. Here in our task, we were trying to figure out whether it's how the brain is responding to those surprises that might be different in PTSD, or whether it's how folks are attending to information once they get that surprise.
And Pearl, do you see a difference? So in the people who have PTSD, does their brain get more surprised more readily than in people who don't?
So the people with PTSD show an equal amount of surprise as people without PTSD, but it's how much attention they're paying to the surprise that's different.
And how are you quantifying that?
So that's what our computational model lets us determine. We have different components of the model that measure how responsive a participant is to surprise, how responsive they are to reward or loss, and how much attention they're paying to the surprise. And in our analyses, we show that it's particularly the attention to surprise that's different in the veterans with PTSD. So everyone shows the same brain response to surprise itself, but it's how much attention you pay to the surprise that extends into choices in the future.
Do you think that that difference was there before they got PTSD, and that's why they're prone to getting PTSD? Or did they develop that as a response to having got PTSD?
Certainly within this study, we can't examine that prospectively, but it is potentially the case that those who are more predisposed to developing these symptoms of hypervigilance might be the ones who are starting off with a predisposition to have this attention-modulated learning process.
And Pearl, what are the consequences then of them paying more attention in this way? The brain generates a surprise signal, it's generic, everyone has it, but then downstream of that, you then end up with, in some people, some areas of the brain responding a bit too much.
So one of the potential consequences is that this contributes to learning difficulties that lead people with PTSD to overestimate the likelihood of negative outcomes into the future, which then contributes to a hypervigilant response.
So does this mean that if a person gets surprised and they then become highly vigilant, Does this transfer to things other than the thing they find traumatic? So will they, for instance, if you present them with, I don't know, a sequence of cards to remember and you surprise them, will they remember those card sequences much better than someone who doesn't have PTSD?
It's a good question. We don't know the answer to that, but it could be the case. A negative outcome, a negative event triggers this increase in attention that changes their focus so that they're learning about really any information that's temporally close to that unexpected negative event. So although we didn't test for that in this experiment, it certainly could generalize to other information that they're seeing in that same context.
And Pearl, does this shed any light on how we can help people with PTSD?
This work absolutely does contribute to how we might treat or understand PTSD in the future. One of our perspectives is that using these computational models that tease out processes allow us to characterize mental illnesses like PTSD in the ways that you might characterize some kind of physical conditions. For example, in the case of hypertension, salt, stress, or smoking may all contribute to hypertension, but depending on which contributes the most, we might have very, very different treatment strategies. So similarly, for PTSD or post-traumatic stress disorder, knowing what contributes in a particular case may help us devise different treatment strategies.
Let's hope so. Pearl Chu and Brooks King-Carsus there from Virginia Tech. One of the largest biological endeavours of recent years was the Human Genome Project that mapped and sequenced all 3 billion of the genetic letters in our DNA code. Now, scientists are embarking on an even more ambitious initiative. From the Sanger Institute, Sarah Teichmann.
We're setting out the goal of mapping all cells in the human body through an international effort that involves cell biologists, genomics experts, computational biologists, and the medical community to collaborate on creating an atlas of the human body at the level of individual cells.
I'm gobsmacked, Sarah. This doesn't exist already. I mean, cell biology has a history going back hundreds of years. So why are we only doing this now?
So it's really thanks to high resolution and high throughput technologies that have been evolving over the past 5 to 10 years that we can now see cells in terms of their entire genomes, transcriptomes, at the level of sort of tens and hundreds of thousands of cells at a time. And so what I'm talking about in a nutshell is the resolution revolution in genomics. This, coupled with also spatial methods that are microscopy based to map cells in their 2- and 3-dimensional context, has meant that we can now grab hold of cells at a completely different level.
It's almost like multi-dimensional then, this atlas, in the sense that, yes, you can say that's what a liver cell looks like, but one can zoom inside the liver cell and say, and this is what it's transcribing. These are the proteins it's making.
That's right. So you're basically describing the entire molecular fingerprint of that cell rather than the morphology of the cell, the way we've been doing with conventional microscopy methods.
Sounds like a huge undertaking, though, because it's not as simple as just saying, I'm going to take a snapshot of an adult now, because obviously cells go through phases during the day, they go through phases during the week. An immune cell can turn from one type of cell to a completely different type of cell as it matures, for example, and we all start life as an egg, which then turns into all these different cell types. So how are you going to do this?
That's a great question, and it really gets to the bottom of asking what is a cell type or what is a cell state. And as you mentioned, there are sort of developmental dynamics. So the human cell atlas will encompass human development and also different ages in the human lifespan. Although in the short term, the aim is to build a first reference initially. So this is a project that we envision will run for a 5 to 10 year term.
I was going to say, it does sound like it's going to be an extremely long-term undertaking. It's huge and potentially bigger than the Human Genome Project because you're going into all these different dimensions.
That's right. It's also more complex at the level of the samples. So basically acquiring tissues from different organs in the body and, as you said, different developmental states. That sample acquisition part is certainly more complex than the Human Genome Project.
I suppose that some of the challenges that had to be surmounted for the Human Genome Project 13 years ago when it finished, before that obviously when it was running, they must be informing or helping with this, because marshalling massive amounts of data was a big challenge then, and it helped to actually change the world and the way that we process data. So you must be able to build on some of that.
Yes, I mean, The Human Genome Project is certainly a template for collaborative efforts in biology and for building a resource. And it's also, as you're saying, the data challenge is similar, although I'd say we're in a new era of big data in biology now where we really are talking serious big data. It's not just a software engineering and database challenge, it also becomes a data mining and statistical and computational challenge with sort of very sophisticated machine learning and artificial intelligence methods that we're developing to make sense of the data and to mine the data and to get the biological knowledge out of it.
There's this quite provocative question which I want to put to you, because I think there will be people listening to this who will think, well, that sounds wonderful, but I don't understand why it matters. Why does having this cell atlas that tells us what's where in the body, but in many dimensions, at many levels, what's going on in those cells, how they're changing and so on, why does this help us do science better?
To me, there's a sort of curiosity argument. We want to know what's inside our body and how it works. But there's also very clear relevance for medicine in the sense of new diagnostics that can be developed based on markers and molecular patterns that we find in the healthy reference body. Immediately it provides a template for saying, okay, what are the cell states that we need to probe that are indicators of disease? Tumor mapping, learning about cancer cell states, essentially having the healthy reference atlas is a very obvious kind of framework for the tumor biology. So there's the basic curiosity about ourselves, but there's also medical applications.
And just in case anyone isn't completely across how massive this undertaking is, put some numbers on it for us. How many cell types? How many different ways of looking at those cell types are you going to have to go across? I mean, what's the scale of this project?
Well, we don't really know how many cell types there are. Conventional estimates on Wikipedia and so on say sort of 200 to 300 cell types, but we're seeing thousands essentially now with these high-resolution technologies. There are 37 trillion cells probably in the human body, and we're not going to sequence all of them, but by sampling in a strategic way from different tissues in the body, we hope to learn about the vast majority of the cell space that's out there. My estimate is that we will sequence billions of cells and we will discover thousands of cell states over the next 5 years or so.
Sarah Teichmann, and I suspect we'll be checking back in with her again in the near future as the results from the project begin emerging. We wish her luck. Meanwhile, that, I'm afraid, is it for this month. You've been listening to the eLife podcast, previous editions of which you can find on eLife's website, that's elifesciences.org, or you can subscribe via iTunes. There are also full text transcripts to each of these programmes, along with the supporting references to the published papers on the Naked Scientist website, which is at nakedscientist.com/eLife. I'll be back with another look inside eLife next month, but until then, thank you for listening, and from me, Chris Smith, goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.